I'm Dr. Greg Mendel, your friendly anatomy professor. Today we're going over chemical digestion lab. So come on in, let's take a look. So first of all, what we're looking at is we are going to digest a protein, a lipid, and a starch. All right. So we have a protein lab where we're digesting from a substrate to a product. In this case, our substrate is a man-made protein called BAPNA. And what this is, made specifically for the experiment, is a group of amino acids with a yellow dye injected into the inside. What we're going to try to do is digest this using an enzyme called trypsin. Now, we know if trypsin is going to digest the BAPNA because this yellow dye will escape if we break down the amino acids. So what we see is hopefully the amino acids will break apart and the product will be a yellow dye and amino acids. So come on in, let's take a look over here. So if you could kind of come in close enough to see this paper. We have 4 test tubes here. In the first and second test tube, we put water and the BAPNA. Now it didn't digest in anything, no yellow, because water doesn't digest proteins. In the second one, we put the enzyme in water. Again, nothing happened. And again, because, well, it's in there. So this one is number 3, and this one's number 4. I tried to grab this first, and what we see is that, I think I had these backwards here. The one that digested a little bit least should be lighter yellow. The one that digests more is a darker yellow. And we see the darker yellow because in the warmth, it should digest more. They mislabeled these two, and I'm sorry. They actually said that, but they put them in the wrong thing. So you can see one is digested least, one digested more. The one more, warmth. All right, let's move to the next thing. So our second lab is called lipids. And so what we're doing is trying to break a substrate down to a product once again. And the substrate that we're using, the beginning thing we're using is called litmus cream. Now in litmus cream, it's just dairy creamer mixed with a litmus powder. And this litmus powder is a pH indicator, not a big deal. You probably saw those little litmus things you put on your tongue, the strips. And if they turn more blue, you're more alkaline. If it's more pink, it's more acidic. Now, the process of digesting this down is really an acidic process. So what we see is this stuff will turn from this purpley color that we start with, and if it digests, we expect it to go a little more pinker colored over here. So we're using an enzyme, in this case called lipase. Lipase breaks down lipids, and we're also, in 2 of these, going to add an emulsifier called bile salts. Remember, the liver makes bile. It's stored in the gallbladder. Emulsifiers take a big chunk and break it into little chunks so that the enzyme could break it down easier. So in the human body and elsewhere, triglyceride is the fat that we're looking at from the dairy creamer, and it is a glycerol backbone with 3 fatty acids. We're trying to break it down in a process called hydrolysis. You might call it hydrolysis. Hydro means water, lysis means break. And we're going to break this down with a water type of bond, but we're going to use lipase to break that. Now, what we hope to end up with is either a monoglyceride, which means there's one fatty acid on the glycerol and 2 fatty acids, or maybe it completely breaks down the glycerol and 3 fatty acids. So let's take a look at what happened here. So come on in so we can take a close look at this paper. I'm going to try to get this as close as possible. But what we see is this is our start. It's litmus cream just with a little bit of water in there. All right, feel free to get to where, there you can see it nice and good. All right, maybe the shadow, I don't know. But the litmus cream, it didn't break it down, obviously. It stayed purple. And then we had water and lipase. So just keep it there, and I was just trying to get out of the shadow. Anyway, so water, obviously, and lipase, nothing. So let's start doing comparisons. All right, so the 2 that were here, and I'm going to sneak in like this, you could see that in heat, it became more pink than this one is not as pink. So what we see is with litmus cream, the lipase broke it down. It became more acidic, therefore turned more pink than this one. The ice did not break down as well as heat. I'm going to switch over to the ones with the bile salts. Now what we can see here is it's still digesting. You can see it's even digesting more at the top than at the bottom. It's working its way down. We just maybe didn't shake it up enough. But this definitely became more pink as compared to this because warmth. I'm going to compare apples to apples now. So I'm comparing the two warm. This has bile salts, and it looks like it's more digested, more pink than the one without bile salts from the original. And then we're going to ice. And what we see is not a lot of difference because it's hard to digest in ice. So we didn't see as much change, even with the bile salts versus non-bile salts in ice. And that is it for the lipid one. Now let's move over to starches. All right, so we're trying to take a starch, we're trying to break it down. The starches are noodles, pasta, what have you, white potatoes. We're using an enzyme called amylase. There's two places in the human body we get this. We get it from the salivary glands, parotid, submandibular, and sublingual salivary glands, puts out salivary amylase. The pancreas dumps out pancreatic amylase. All right, so those two things should break it down into smaller pieces. Now, when I say smaller pieces, the starch is a polysaccharide. It's a long chain of sugars, right? Long chain of sugars forms a starch in a vegetable. And we're trying to break it down into a disaccharide, which is smaller, but it's still two pieces together. We have three well-known disaccharides in the human body. We have lactose, maltose, and sucrose. The three things that break it down is lactase, maltase, and sucrase. Those would be the three enzymes that are brush border enzymes. But in this case, we're going to try to take amylase and break these down. In the human body, again, lactase, maltase, and sucrase. We're going to try to break a disaccharide into a single sugar. Some are hexagonal shaped, some are more pentagonal shaped. But what we see is a lactose, when we break it down, changes into one glucose and one galactose. When we break down a maltose, it breaks down into two glucoses. When we break down sucrose, it breaks down into a fructose and a glucose again. Glucose is what the body loves as sugar. So we're trying to break this down best we can to come up with as many glucose as we can. Now, how do we know what changed? All right, the first thing we're going to do is an experiment where we use a solution called IKI/Legules. That is a reagent, and it tests for the presence of starches. So it is gold in color, but when you put it near a starch, it changes black. All right, so if you use IKI/Legules reagent, which is gold, in the presence of starches, it turns black. That's one test. So if it stays gold, we know that either there was not starch or all the starch completely digested into glucose, so there's no starch left, or it just didn't have any in there. But when it does digest, we expect it to turn into, again, one of the products, glucose, sucrose, or galactose. Benedict's reagent, which is blue, light blue, if we heat it, it should change any other color other than blue if these sugars are present. So we did both of those. Come on in, let's take a look. All right, so what we're looking at here is we're taking these starches, the first, second, third, fourth, and fifth, and what we did is we mixed water and amylase in the first one. Well, there's no starch, that's a control group, so it's gold. Second one, water and starch. Well, we do have starch. We're testing for starch with IKI/Legules. Very black. The next one, water and glucose. Gold, because again, no starch, it's a control. Now here's our experiment. We're making the starch, mix it with amylase, and we can see if it digests. What we saw earlier is that this one had digested more than this one. And it still kind of appears that way. It's lighter than this one, which means some digestion happened. Some starch converted to a sugar. So it tests not nearly as dark as the pure starch, but it's digesting to sugar. Therefore, it's not black and not gold, it's halfway there. This one's darker because it was in cold and it just does not digest as well. So now let's take the Benedict's reagent and run the test. So what we saw here is we have, and again, we're testing for sugar. Water and amylase, water and starch. Neither have sugar. It stayed light blue. Next, we have our control group. This is here. Water and glucose changed very orange. And then we have these two. Now, what we see is they're about the same color because we just boiled both of them. So it doesn't matter if it's 37 or 0 at this point. They tested positive because there was some digestion happened. Starch gets digested by amylase. It changes the sugar. This went no longer blue, digestion did occur. And that is it for our video.